Hi, welcome to our 5th AP Biology lesson. Today our lesson will be on new material in Unit 7. On Friday, April 3rd, an announcement will be made providing details about the 2020 AP Biology exam. Beginning on April 7th, we will be presenting lessons that will review the first 6 units in the AP Biology course. All lessons, including these with new material, will include practice with science skills, which are relevant with any content. We will also provide practice with free response questions in each lesson. My name is Allison Kate. I'm affiliated with both UC Berkeley and Redwood High School, and today I'm teaching you from my home in El Cerrito, California. So we're going to start today with a warm-up. And in case you're just joining us, these warm-ups are presented as homework in the previous lesson. So this is homework from yesterday's lesson, topic 7.4, which is about population genetics. And so let's take a look at the question. It's got a data table, which is titled Table 1, the effect of 0.1 millimolar caffeine on memory in bees. And you can see that in the far left column, there is the independent variable or treatment, which is caffeine, and it is compared to a control. And then we see the dependent variable is memory, which is measured by the average probability of revisiting a nectar source plus or minus 2 standard errors of the mean. And you can see that that data was collected at 2 time periods, 10 minutes and 24 hours. So now let's look at the question. And if you're just joining us today, please grab a pen and paper and you can try to jot down an answer as I'm talking about this, but I will be debriefing it in the next slide. In flowering plants, pollination is a process that leads to the fertilization of an egg and the production of seeds. Some flowers attract pollinators, such as bees, using visual and chemical cues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. Caffeine is a bitter tasting compound that can be toxic to insects at high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effect of 0.1 micromolar caffeine on bee behavior. The results of this experiment to test the effect of caffeine on bees' memory of a nectar source are shown in Table 1. And that's what I started out discussing with you. So you are asked in this question to design an experiment using artificial flowers to investigate the potential negative effects of increasing caffeine concentrations in nectar on the number of floral visits by bees. In that experimental design, you are asked to identify the null hypothesis, an appropriate control treatment, and the predicted results that could be used to reject the null hypothesis. And also you were asked to make a graph of this data to give you more practice with that skill. So if you did this last night, grab your homework. And if you're just joining us today, try to think about your experimental design. You might not have time to actually do the graph, but you can go back and do that later. All right, so the scoring guidelines for this question show that your experimental design piece was worth 3 points, and each row would have earned you 1 point. So the null hypothesis in this particular case is that increasing caffeine concentration has no effect on the number of floral visits by bees. The control would be nectar or flowers with no caffeine. And the predicted results, you could have earned a point with either of those bullet points. The number of floral visits by bees is different at increasing caffeine concentrations, or the number of floral visits by bees is different than the control. So each one of those parts of the experimental design was worth 1 point. Now let's take a look at the graph. I hope you did it. If you didn't, you should practice this because this is an important skill that can be applied to any content throughout the course. I have 2 images here because you could have earned full credit for either one of these ways of setting this graph up. So let's take a look at the graph on the left first. You can see that the dependent variable on the y-axis is memory, or the average probability of revisiting the nectar source. And then the x-axis is the independent variable, and it is the treatment. And the first student showed both the control and the caffeine next to each other at 10 minutes, and then the control and the caffeine treatment at 24 hours. You can also see that the student correctly used the plus or minus 2 standard errors of the mean by showing those error bars. And there's a lovely title on this graph as well, the effect of caffeine on memory of bees. So that was a full point question, which is 3 points for the graph. So just a little variation that still would have earned full credit. If you look at the graph on the right, we have the same dependent variable, probability of revisiting the nectar source. This student chose to display the data a little bit differently. We still have the time at 10 minutes and 24 hours, but the treatments are shown as either shaded or open. So the shaded is the control and the open is the caffeine. And there is a key to indicate which is which. And again, this student also has error bars done correctly and the title. So both of these graphs were full credit graphs. All right, so today's lesson overview. First thing, don't forget your pen and paper. They're going to be used today, and today you need a calculator. So you can use your phone or you can get your calculator out. The concept today is Hardy-Weinberg Equilibrium, and the science practice skill is 5AA, which is performing mathematical equations, including mathematical equations in the curriculum. And we're going to gain proficiency by looking at sample multiple choice questions requiring the use of the Hardy-Weinberg equation. We're also going to look at FRQ practice questions that combine the content and skill. So as we embark on this lesson today, you should be familiar with the concepts of population genetics. My colleague, Ms. Pfizer, presented a lesson on that yesterday. You should also be familiar with the terms allele, genotype, phenotype, and frequency. And by the end of this lesson, you will be able to do the following. You should be able to describe the conditions under which allele and genotype frequencies will change in populations. You should be able to explain the impacts on the population if any of the conditions of the Hardy-Weinberg are not met. And you should be able to perform mathematical calculations using the Hardy-Weinberg equation. So here's what you need to know. This is just to pinpoint where we are in the large AP Biology curriculum to our lesson today. So we are currently working in Unit 7, which is about natural selection. The units are broken into small topics. Each topic has a learning objective, and this is what the students need to be able to do with your content knowledge. And the essential knowledge is the knowledge required to perform the learning objective. So today we are working on topic 7.5, which is the Hardy-Weinberg Equilibrium. There are 2 learning objectives that we're working on. The first one is to describe the conditions under which allele and genotype frequencies will change in populations. And the second is to explain the impacts on the population if any of the conditions of Hardy-Weinberg are not met. The essential knowledge, which is the knowledge required to be able to do those learning objectives, is that first, the Hardy-Weinberg is a model for describing and predicting allele frequencies in a non-evolving population. And there are 5 conditions for a population to be in Hardy-Weinberg equilibrium. So the last thing today to remember is that today's topic skill pair is Hardy-Weinberg equilibrium and performing mathematical equations. All right, so a little background about the Hardy-Weinberg model. Hardy and Weinberg were 2 different people. Hardy was an English mathematician and Weinberg was a German physician. And independently, at the same time, they both worked out the effects of random mating in successive generations on the frequencies of alleles in a population. So we give both of these men credit for coming up with a model that describes these effects. This is important for biologists because it's the basis of a hypothetical stability from which real change can be measured. So it is the null hypothesis. It is when a population is not evolving so that we can use it to compare an evolving population to. So this model also allows us to figure out the frequency of genotypes from phenotypes and to track any changes in allele frequency, which means evolution is occurring. So the Hardy-Weinberg model again describes a hypothetical situation. It serves as the null hypothesis, which in this case is a non-evolving population. The model is for when all agents of evolutionary change are absent. So I'm going to go through these, and then in the next slide, I'm going to present what those agents of evolutionary change are. So you can see it from both sides. So in the model, there can be no genetic drift, and that means no small populations, so you have to have a large population size. There can be no gene flow, which means no migration of alleles into or out of a population. There can be no mutations, which are changes to the DNA. Mating must be random. There can be no sexual selection, no mate preference, and there can be no natural selection, which means that there's equal survival rates. And again, this is a model. This seldom occurs in natural populations, which is why we can compare what happens in natural populations to this model. So here are the 5 agents of evolutionary change. So let's take a look at each one of these. The first one is mutation, and a mutation is simply changes to the DNA. The second one is gene flow, which is migration. The third one is non-random mating. There can be no sexual selection. Genetic drift means that you can not have small populations, and then selection, which means that there is differential survival of individuals in the population. So putting it all together, remember, the 5 conditions required for Hardy-Weinberg equilibrium to hold true are a large population, in other words, no genetic drift, isolation from other populations, no gene flow, no net mutations, mating must be random, and no natural selection. Remember these. Okay, you just have to remember these 5 conditions, and you have to remember that these conditions are seldom met in natural populations. So let's take a look at the math. We're going to take a look at the Hardy-Weinberg equations. And these equations are from the AP Biology equations and formula sheet found in Appendix 5.1 in the AP Biology course and exam description. This is also available to you on exam day. So there are 2 equations that I'd like you to get familiar with. We're going to practice with these in the next several slides. The first equation says p squared plus 2pq plus q squares equals 1. Okay, so P represents the frequency of one allele in a population, and Q represents the frequency of the second allele in a population. The first equation, P squared plus 2pq plus q squared equals 1, is used to determine genotype frequencies. And the reason we have equals 1 is that all of those genotypes have to add up to all of the genotypes in the population. The second equation is P plus Q equals 1. This is a really useful equation. These represent the allele frequencies, and all of the alleles have to add up to 1. So kind of the cool thing about Hardy-Weinberg is you can always check your math. If you get some weird number and it doesn't add up to 1, you know you've done your calculation incorrectly. So a couple things to remember about the components of this equation. To calculate allele frequency, you use P plus Q equals 1. P represents the dominant allele, and that's usually symbolized with a capital letter. Q represents the recessive allele, and that's symbolized with a lowercase letter. To calculate the genotype frequency, you use the P squared plus 2pq plus Q squared equation, where P squared represents the homozygous dominant genotype, and 2pq represents the heterozygous genotype, and Q squared represents the homozygous recessive genotype. Don't worry, we're going to practice with this. So let's take a look at how the Hardy-Weinberg equilibrium, how Hardy and Weinberg actually kind of thought about this. So there are 2 images. Let's look at the image on the left. and we're looking at some flowers, and we're looking at phenotypes. And you may recall from genetics that there are two ways to have the dominant phenotype. The first way is to be homozygous, right? So you're homozygous dominant, which is displayed or represented by capital A, capital A. And you can also have the same phenotype if you're heterozygous, because as long as you have one dominant allele, you express that phenotype. So there are two ways to be a pink flower. The white flower is the homozygous recessive. So let's say that we have got a population of plants where the total number of plants in this population is 500. And we know that 320 of them are homozygous dominant. Maybe we did a test cross to figure that out. And 160 are heterozygous and 20 are homozygous recessive. So if you have the total number of plants and you know the genotype frequencies, you can figure out the frequency of each genotype. I'm sorry, I said that wrong. Let me check that. If you know the total number of plants and you know the total number of a particular genotype, then you can determine the frequency of each of those genotypes. So in the first case, the homozygous dominant genotype would be 320 out of 500, which is 0.64. Now you can look at the heterozygote. The heterozygote is 160 out of 500, or a frequency of 0.32. And the homozygous recessive is 20 out of 500, or a frequency of 0.04. If you look at the number of alleles, you have to remember that each individual has two alleles. And so what happens is when you're figuring this out, and you'll see this when we get to the heterozygote, you have to multiply the number of alleles by 2. So we had 320, multiply that by 2, and that's 640 dominant alleles out of the total number of alleles in the gene pool, which is 500 times 2, or 1,000. And so now we get the allele frequency of the dominant allele is 0.8, and that is represented by P in Hardy-Weinberg language. Now let's look at the heterozygote. Remember, the heterozygote has half, 160, of the dominant allele and half, or 160, of the recessive allele. So that is where we get the 800. We had 640 from the dominant genotype, and then 160 from the heterozygote. We look at the recessive allele, little a, which is represented by Q in Hardy-Weinberg language. We have 160 of those recessive alleles from the heterozygote and 40 from the homozygous recessive. Add those together, and that's 200 out of the total number of alleles, 1,000, and you get your frequency of 0.2. And again, the beauty of Hardy-Weinberg is you can check your math. 0.8 plus 0.2 equals 1. That's all of the alleles in the population. The image on the right just shows why that 2PQ comes about. If you take the gametes from the parents, you see the egg and sperm, and you can see that there's two ways to get that heterozygous genotype. There's little a, big A, and there's big A, little a. So that's where the 2PQ comes from. Okay, some things to know about the Hardy-Weinberg equation. The convention when solving Hardy-Weinberg problems is to use decimals to show the frequencies. You may see coming up some questions that ask you to determine the percent of a genotype or an individual. And since we're using decimals, all you would need to do is multiply by 100 to get that percent. And we'll get some practice with that in a minute. Knowing that P plus Q equals 1 allows you to determine the other allele value if you know 1. For example, if P is 0.7, then Q is 1 minus P, or 1 minus 0.7, Q equals 3. Add them up, 0.7 plus 0.3 equals 1. And my personal favorite is when you begin to solve a Hardy-Weinberg problem, you always want to start with solving for Q, or Q squared, because you know the homozygous recessive phenotype. If you know the phenotype, you know it has to be the genotype, because with those homozygous recessives, what you see is what you get. All right, so let's get some practice. I'm going to go through a couple of problems and then I'm going to let you practice on your own. Okay, so the key takeaway for this first one is that P and Q represent the allele frequencies of the dominant and recessive alleles. Let's look at a practice problem. The allele, capital W for white wool, is dominant over the allele, lowercase w for black wool. In a sample of 900 sheep, 891 are white and 9 are black. Calculate the allele frequencies in this sample. So you always want to begin with what you know, and then you need to figure out what you're solving for. So they're asking us to calculate allele frequencies. So we know that there are 891 out of 900 white sheep. Remember, we don't know if they're homozygous dominant white or heterozygous white. So we don't want to start there. But we also know that 9 out of 900 are black sheep. And since black is recessive, we know that 9 out of 900, or 0.01, is the homozygous recessive genotype. So that's Q squared. So if Q squared is 9 out of 900, then Q has to be the square root of that, or the square root of 0.01. So Q equals 0.1. Now that we know Q, we can easily determine P because P equals 1 minus Q. So P is 0.9. So here's our answers. The frequency of the dominant allele P is 0.9, and the frequency of the recessive allele Q is 0.1. And again, you can check your answer by adding those together, and they equal 1, or all of the alleles in the sheep population. So let's do the same thing, but this time let's look at genotype frequencies. Your key takeaway is that P squared, 2PQ, and Q squared represent the genotype frequencies of the homozygous dominant, heterozygous, and homozygous recessive genotypes. So let's do one more together. In a given Hardy-Weinberg equilibrium with allele frequencies where the dominant allele capital A is 0.6 and the recessive allele lowercase a is 0.4, determine the frequencies of the 3 genotypes. So again, begin with what you know. Since we're solving for the genotype frequencies, we must use the correct formula, which is P squared plus 2PQ plus Q squared equals 1. Since we're given the frequency of both of those alleles, we know that the dominant allele P is 0.6 and the recessive allele Q is 0.4. We just need to plug them into the equation. So for P squared, it's 0.6 squared. For 2PQ, it's 2 times 0.6 times 0.4. And for Q squared, it's 0.4 squared. So our genotype frequencies are 0.36 for homozygous dominant, 0.48 for the heterozygote, and 0.16 for the homozygous recessive. And again, if you add those frequencies together, you get 1, or all of the genotypes in the population. So now you're going to try it. All right, get that calculator, get your pen and paper handy. In a population that's in Hardy-Weinberg equilibrium, the frequency of the Z allele is 0.6. What percentage of the population is heterozygous at this locus? So first of all, you're solving for the heterozygous genotype. You're given that one allele is 0.6. And this is a percentage problem. So whatever your final answer is in decimal form, you need to convert to percentage by multiplying by 100. Okay, so take a minute and solve this problem. Don't worry, I'll go over the answer with you. Okay, I know that that's not a lot of time, but let's move on. So you know that one allele, Z equals 0.6. So that means you know the other allele because P plus Q equals 1. So your allele frequencies are 0.6, which was given, and 0.4, which was easy to determine. Probably didn't even need to use your calculator for that one. And now you're asked to solve for the heterozygous genotype. So you're plugging in 0.6 and 0.4 into the 2PQ part of the genotype frequency equation. So that 2 times 0.6 times 0.4 is 0.48. And because the question asks for percentage, you simply multiply 0.48 by 100, and your answer is, 48% of the population is heterozygous. Hope you got that answer. All right, let's do a couple more practice questions in just a minute. I wanted to bring you back into the classroom because if you were in the classroom, your teacher may choose to do a lab investigation on this topic. And I'm going to start with the AP Bio Lab Investigation 2 from the College Board lab manual. And that is a mathematical modeling activity in which you use Excel to model Hardy-Weinberg allele frequencies. In this case, you can build your own Excel spreadsheet and then play with the variables to look at how those frequencies change over time. Or there are some already built spreadsheets that allow you to change the parameters and see how those allele frequencies change. Another one is called fishy frequencies, and this one is done using goldfish crackers and a prey-predator simulation to model the null hypothesis, which is no selection, and then evolution, which is selection against one of the goldfish phenotypes. And the fun thing about this activity is you are the predator, so you get to select against one of those goldfish cracker phenotypes. This can also be done with any two different phenotypes. So you could use animal crackers, or you could use M&Ms of two different colors. It's fun because you get to eat your data, which is always a good thing. And then finally, Rock Pocket Mouse has an activity that uses authentic data. And let me just remind you that in our very first lesson, we showed a little video clip of the Rock Pocket Mouse. And this is a modern day example of how protective coloration is a selective advantage. And this investigation uses that actual data that was collected by Dr. Michael Nachman to determine the frequencies of the light and dark pocket mice in a population over time. And this is done right in the southwestern desert of the United States. One little personal note, turns out that Dr. Nachman and I were classmates back in the Paleozoic era at UC Berkeley. We took a vertebrate zoology course together. There was a recent reunion, so I was kind of a fangirl and got my picture taken with him. So that was pretty cool. All right, so any of these three are labs that you could do to investigate the Hardy-Weinberg equation. All right, so let's do a check for understanding. We're going to really focus on the skill of performing mathematical equations, including the mathematical equations in the curriculum. And I just can't stress this enough. If I were in my classroom with my students, I would just tell them that the key to Hardy-Weinberg problems is to practice, practice, practice. So let's practice some more so we can master this skill. This question tells you that 9% of an African population is born with a severe form of sickle cell anemia. So this is the homozygous recessive genotype, or s, which is also known as Q squared in Hardy-Weinberg language. The question is asking what percentage of the population will be resistant to malaria because they are heterozygous, big S, little s, for the sickle cell gene. And this is something that you'll see over and over in AP Bio. This example of sickle cell anemia comes up in a variety of content topics. But in this case, the folks who are heterozygous for this particular allele do have protection from malaria. So in solving a Hardy-Weinberg problem, you know that 9% represents Q squared. It means that Q squared is 9 out of 100 because it's percent, or 0.09. So Q is the square root of 0.09, or Q is equal to 0.3. Once you know Q, you can solve for P. Again, you probably don't need your calculators. P is 1 minus Q, or 0.7. And now we just plug those values of P and Q back into our equation for the heterozygous genotype, which is 2PQ. So 2 times 0.7 times 0.3 is 0.42. Now, here's what you got to watch out for. Remember that the question asked you what percentage. So you don't want to leave that in decimal form. You want to convert that to percent by multiplying by 100. So your answer is 42%. All right, now you get to try. And I have 3 slides for you. They're all multiple choice Hardy-Weinberg problems. You need that calculator, and I want you to try these on your own. Don't worry, I'll provide you with the correct answer so you can check your work. And I'll also not just tell you it's answer choice B, but I'm going to show you how that works. Are you ready? All right, so here's your first practice. A particular gene has 2 alleles, a dominant allele, capital A, and a recessive allele, lowercase a. The frequency of the dominant allele is 0.55. If the population is in Hardy-Weinberg equilibrium with respect to the gene, then what is the expected genotype, expected frequency of genotype big A, little a? Okay, so you're solving for 2PQ. Got it? Okay, in this case, the correct answer was D. You all got that, right? And here's how we came up with that correct answer. We are solving for big A, little a, or 2PQ. We know that for big A or P, it's equal to 0.55. They gave that to us in the question. So Q has to be 0.45 because we know that P plus Q equals 1. So now we just put those values in to the 2PQ part of the equation and solve the question. 2 times 0.55 times 0.45 equals 0.495. And again, it's kind of a weird number. Don't let that throw you off because those values add up to 1. Okay? Ready for the next one? Might be a little bit harder. All right, so Gaucher disease or type 1 or GD1 is a recessive genetic disease that affects 1 in 900 individuals in a particular population. GD1 is caused by mutation in the enzyme glucocerebrosidase. I'm sure I didn't say that right. Assuming the population is in Hardy-Weinberg equilibrium, calculate the frequency of the wild type or non-mutant allele for the enzyme. I'm not going to try it again. Glucocerebrosidase. Awesome. Okay, so now that I've massacred the enzyme name, you are calculating the frequency of the non-mutant allele, and you are given the recessive genetic disease affects 1 in 900 individuals. All right, go. If you need a hint, think about what they're giving you in the question. Okay, so that one's a little harder, not much, but let's take a look at the answer. So in this case, the correct answer is C. They gave us Q squared in the question. So remember how my advice was to always start with Q or Q squared? They gave us Q squared. So we know that 1 out of 900 or 0.00111 is equal to Q squared because GD1 is a recessive genetic disease. The question is asking us to solve for P, which is the non-mutant allele. And we start by finding Q, which is the square root of Q squared, right? So it's the square root of 0.00111 or 0.033. And now we can solve for P because we know that P plus Q equals 1. So P equals 0.967 or answer choice C. All right, one more. Okay, so humans vary in their ability to taste the bitter chemical compound phenothiocarbamide, PTC. The taster phenotype is dominant to the non-taster phenotype. Researchers tested the PTC taster phenotype of individuals from an isolated population. In the sample, 780 individuals were able to taste PTC and 70 individuals were not able to taste PTC. Assuming the population is in Hardy-Weinberg equilibrium, what is the frequency of the non-taster allele. Again, think about what they're giving you. Think about what they're asking you to solve for. Do your math and come up with an answer choice. Okay. So this question asked us to solve for the non-taster allele, so we're solving for Q. We also know that 70 out of the total number of individuals, which is 850, so 780 plus 70 is 850, are equal to Q squared. So Q squared is equal to 0.082. So to get this answer, all we have to do is take the square root of 0.082, and that is 0.286 rounded up to 0.29, and that's answer choice B. So once you get the hang of these, they're pretty fun. I think they're pretty fun. Practice, practice, practice with Hardy-Weinberg problems. All right, so our debrief and summary, you should now be able to describe the conditions under which allele and genotype frequencies will change in populations. You should be able to explain the impacts on the population if any of these conditions of Hardy-Weinberg are not met, and you should be able to perform mathematical calculations using the Hardy-Weinberg equation. Your key takeaways is that evolution is characterized by a change in the genetic makeup of a population over time and is supported by multiple lines of evidence. In this case, evolution is supported by a change in the allele frequency. Hardy-Weinberg is a model for describing and predicting allele frequencies in a non-evolving population. So it is only a model that we can compare natural populations to see if they are evolving. So let's talk about some common errors and misunderstandings, and also just some reminders for how to use this and how to solve Hardy-Weinberg problems. Each individual has 2 alleles for any given gene, one from each parent, so that the total number of alleles in a population is the number of individuals times 2. We had some practice with that in the last practice, multiple choice practice question. And also, if you go back to the graphic I showed you of how this was derived with the flowers, it really illustrates that nicely. You cannot calculate genotype or allele frequencies from phenotype alone unless you know the phenotype of the homozygous recessive. In this case, what you see, the phenotype, is the genotype, and often they will give you that in the problem. If you're given the number of individuals with particular phenotypes, you can only be sure of the genotype of the individuals with the recessive phenotype. So start by figuring out Q squared and Q before anything else. You cannot start knowing the phenotype of the dominant homozygous dominant because you don't know if it's homozygous dominant or if it's heterozygous. And I hope that makes sense. So you cannot use Q squared or any other like the P squared or the 2PQ to determine the allele frequencies. You have to count the alleles to get allele frequencies. So I'm going to leave you with these. You need to know and tell the difference, don't mix them up, between allele and genotype frequencies. In other words, mind your P's and Q's. Single letters such as Q, P, R, etc. represent allele frequencies. And remember, there could be more than 2 alleles for a particular gene. The allele frequency then is equal to the number of a given allele divided by the total number of alleles in a population. So that's allele frequencies. Any letter that is not a single letter, like P squared, Q squared, R squared, 2PQ, represents the genotype frequency, which is equal to the number of individuals with this phenotype divided by the total number of individuals. You can only count the alleles if you know the number of individuals with a given genotype. So that's a lot of try to remember, mind your P's and Q's, must knows. Again, once you start solving a lot of these problems and you practice, these will make a lot of sense. Okay, so let's give you some AP exam practice. This is your homework for tonight, and Ms. Pfizer will go over this with you as your warm-up tomorrow. So this is from a FRQ from a previous exam, and it reads, evolution is one of the unifying themes of biology. Evolution involves change in the frequencies of alleles in a population. For a particular genetic locus in a population, the frequency of the recessive allele, little a, is 0.4, and the frequency of the dominant allele is 0.6. You're asked to do 3 separate things. In part A, what is the frequency of each genotype? So you need to calculate the genotype frequencies for big A, big A, little a, and little a in the population. And then you need to calculate the frequency of the dominant phenotype. Part B, how can the Hardy-Weinberg principle of genetic equilibrium be used to determine whether this population is evolving? And in part C, you're asked to identify a particular environmental change and describe how it might alter allelic frequencies in this population. And I'll give you a hint. Think about the conditions that have to be present for Hardy-Weinberg to hold true. And I would set the timer for about 20 minutes as you work through this problem. Remember, if you were given this problem on the AP exam, you would have the formula sheet and you would have a calculator. So that takes that stress away. All right, so this is your homework for tonight, and it will be debriefed tomorrow as your warm-up for the next lesson. One more thing before I say goodbye today. We know that not all students have access to the internet or to a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach out to us directly and let us know. And you can do that at cb.org/tech. I'd like to thank you for joining me today. I hope you'll join us for all of our lessons. Be sure to complete that practice question because we're going to review it in the next lesson. Have a great rest of your day.